Hello, welcome to the first lecture discussion for Pmls 1. Today's topic is History of Medical Technology Profession. Now, before we begin, allow me to state the objectives for this topic. At the end of the period, the students are expected to discuss the history of medical technology in a global context, identify the important personalities that played significant roles in the progress of the medical technology profession, and to cite inventions and innovations in the medical technology field. Now, what is medical technology? How do we define it? There has been several contributors to the definition of medical technology. Let's take a look at who they are. Anne Fagelson defined medical technology as a branch of medicine which is concerned with the performance of laboratory determinations and analysis used in the diagnosis and treatment of disease and the maintenance of health. Another contributor, Ruth Heinemann, defined medical technology as an application of the principles of natural, physical, and biological sciences in laboratory procedures to aid in the diagnosis and treatment of diseases. Another contributor named Walters defined medical technology as a profession concerned with performing laboratory analysis in view of obtaining information necessary in the diagnosis and treatment of diseases, as well as in the maintenance of good health. Now, the best definition for us here in the Philippines would be that which is given by RA 5527, or the Republic Act of 5527, which is the Philippine Medical Technology Act of 1969. According to this Republic Act, medical technology is a profession which is usually concerned with examination of tissues, secretion, and excretion of the human body and body fluids by various electronic, chemical, microscopic, and other medical laboratory procedures or techniques, either manual or automated, that is used to aid the physician in the diagnosis, study, and treatment of disease, and in the promotion of health in general. Now, medical technology is actually an interdisciplinary study wherein those that would like to go into that profession should train in different disciplines such as hematology, clinical chemistry, medical microbiology, clinical microscopy, immunology, serology, and general pathology. Now, to understand how medical technology today came about, we should take a look at its history of how it came to be. Now, the earliest book or text showing explanations about medicine or parasites or diseases was the Ebers Papyrus, which was written about 1500 BC. It is the oldest preserved Egyptian compilation of medical texts that contains many incantations meant to turn away disease-causing demons, and there is also evidence of a long tradition of empiricism. It is actually about a 110-page long scroll, or about 20 to 23 meters long. It shows chapters on contraception, pregnancy, eye and skin problems, surgery, burns, and intestinal diseases, as well as parasites. Vivian Herrick, an early scientist, traced the beginning of medical technology to that particular date, about 1500 BC, when intestinal parasites such as Taenia and Ascaris were first identified from mummies or dead bodies exhumed in Egypt. Hippocrates, another contributor to the science of medical technology, is considered as the father of medicine. He advocated the use of mind and senses as diagnostic tools. He even described 4 humors or body fluids in the human body. Today, Those theories about humors is called Central Hippocratic Doctrine of Humoral Pathology, which states that all diseases were due to the disorder of these four humors or body fluids. Now, what are these four humors? Looking at this illustration at the side, the four humors are known as 1. Yellow bile, 2. Black bile, 3. Phlegm, and 4. Blood. These are said to be associated with different diseases and temperaments. Okay? When there's increase or decrease or imbalances in these humors, there is a particular disease condition that is associated with it. Say, for example, if there's increase in yellow bile, the patient is said to display a choleric type of temperament or attitude. Choleric means the person is almost always angry or irritable. That means that person is sick somehow in his liver. There's something wrong with his liver. Or if there's increase in black bile, the patient is said to display a melancholic type of temperament, or he is almost always sad or depressed. There's something wrong with his spleen. Okay? Now, phlegm, if there's increase in phlegm, which is said to be produced by brain and lungs, okay? A patient that is shows an increase in phlegm is said to display a phlegmatic type of personality. Phlegmatic means unfeeling, okay? Stoic, or poker faced. Okay, and then if there's increase in blood, no, the patient is said to display a sanguine type of personality. He's almost always friendly. The complexion rather is ruddy or almost always pink. Okay, but there's something wrong with his heart if that is the case. Okay, so that's how they diagnose diseases rather during the time of Hippocrates. Okay, now during these times, urine was regarded as composite of these humors. So they test urine and check for the presence of yellow bile, black bile, phlegm, and blood. However, that is not how we do it at this point of time. Now, during those times, urinalysis is considered as a prime mode of diagnosis. So it is considered as the oldest laboratory procedure. The urine flask is a symbol of medieval medicine. Now, Greeks and Hindu doctors at that time also made scientific observation, open and closed quote, with urine. Now, they tried to spill urine on the ground and observe it if it attracts ants. If it does, it means the urine is sweet. So that was how they diagnosed diabetes at that time, although they were still not aware of that particular term. The Chinese started with immunization quite early, earlier than what we understand immunization to be. And during those times, smallpox was a disease that ravaged a lot of the areas in China. And they tried to acquire immunization by powdering smallpox scabs. So they take off smallpox scabs from patients and powder it, and then they put it at the tip of a long straw and blow it into an individual that they would like to be immunized from smallpox. So looking at the illustration below, so the physician here is blowing powdered smallpox scabs into the nose of a young boy. That is how they tried to immunize individuals at that time. Although the results is a 50-50. Some acquired immunizations while others actually suffer from the infection thereafter. Then we have Rufus of Ephesus, which made the first description of hematuria. Hematuria is the presence of blood in urine. So as early as 50 AD, they were actually aware of problems in the kidney wherein it is not able to filter blood anymore and reuse it for the body. Then for the Middle Ages, around 500 to 1500 AD, a contributor to the science of medical technology was a Jesuit scientist, Jewish scientist called Isaac Judaeus. He is considered to be one of the founders of the origins of nephrology. He created a book entitled Kitab al-Baul, which literally translates to Body of Urine. It's a book which is a compilation of the guidelines for the use of urine as a diagnostic aid. So you see, so as early as 500 AD or way, way earlier, they actually practiced urinalysis. Then we have Zacharias Janssen. No? This individual, together with his father Hans, invented a simple microscope during the 1590s. Okay? There's actually no details or illustrations as to how his um, microscopes look like. Then we have Ruth Williams. Ruth Williams believed that medical technology began in the medieval period at around 1096 to 1438 because of the fact that urinalysis was a fad during that time. Anne Fagelson again believed that medical technology started in the 14th century, however, when an Italian doctor at the University of Bologna employed Alessandra Giuliani to perform different tasks in the laboratory, like examination of urine and so on. And during the 15th century, this century saw to the discovery of different dyes, such as aniline dyes that were used in staining microorganisms. Now, moving on to laboratory practices in the 16th to 18th century. So a prime contributor in this age was Anton van Leeuwenhoek. Leeuwenhoek is actually considered to be the father of microbiology. He is known to be the first scientist to actually see the three basic shapes of bacteria: spherical or round, rod-shaped or bacilli, and the spiral bacteria. He was only able to use tooth scrapings and canal water to do this discovery. However, he was quite lucky to have seen those shapes. He was so excited. To him, they looked like tiny beasts. He called them animalcules. They were moving around since he tested canal water, so there were living organisms in the sample. And he was able to see that using just this simple microscope that he invented. Okay? So it's a single lens microscope that you actually have to hold and move close to your eye. Now, not until some time later, when Robert Hooke commissioned a friend to create a special type of microscope, that the discovery of Anton van Leeuwenhoek was actually acknowledged. Okay? So Robert Hooke, who again commissioned a friend to make a special type of microscope made out of wood and metal and leather, he used that microscope to look into structures of tiny microorganisms and skins or peels of fruit and vegetables and bark of oak trees. And he compiled into a book called Micrographia, everything that he saw under his microscope. So he was a very talented sketch artist. So he drew everything. Okay, apart from that contribution, he actually coined the term cell. So looking at an oak bark, he was able to see the tiny square-like structures that make up the bark. And those tiny structures mimic the cells of priests or abbots in an abbey. Okay, so the rooms of those priests were called cells. Okay, so he used that term to call those square-like structures that make up the oak bark that he sampled for his book. Apart from that, he also contributed quite a number of theories in mathematics. Okay. Then we have Athanasius Kircher. This scientist is considered to be the greatest of the early microscopists. In his experimentations, he was actually able to see blood, and he tried and experimented with blood of patients that were sick with plague at that time, not the Black Plague. So in his discoveries, he mentioned that the blood of plague patients contained worms. Okay? But when we take into consideration the type of technology that they have during that time, you might not have been able to see the bacteria that would have caused the plague, but he might have had seen uh, blood cell components, like RBCs and WBCs, and perhaps some platelet. They'd be moving around if there's draft in the area. Then we have Marcello Malpighi, who was an Italian microscopist. He had several contributions in embryology and anatomy. And because of that, he was considered to be the founding father of modern anatomic pathology. Then we have Jean-Baptiste van Helmont. This scientist introduced the gravimetric analysis of urine. Richard Lower tried to investigate blood transfusion from one animal to another, and he was actually quite successful. Then we have Frederick Dekkers. Dekkers was a Dutch scientist who observed that proteins in urine precipitated when boiled with acetic acid. And thanks for that discovery and observation, we actually use that type of experimentation in our biochemistry up until today. William Hewson, on the other hand, discovered that plasma could be separated from blood cells when blood clots. Okay? So since there is still limited knowledge about blood clotting during his time, he called the liquid portion of clotted blood as plasma, when in fact it is serum. Okay? Plasma is the liquid portion of unclotted blood, and serum is the liquid portion of clotted blood. Then we have Edward Jenner. Okay, Edward Jenner is known for his contribution in vaccination and immunology. Okay, he inoculated a person, a child actually, with cowpox virus, resulting to protection from smallpox. So that protection is called immunity. Yes, he's actually quite a curious scientist, and noticed that some of his neighbors or some of the individuals around his area were not suffering from smallpox, unlike the rest. Yeah? So he tried to question those individuals that were unaffected by smallpox about the infections that they had previously, if they had smallpox before. One lady, a milkmaid, said that she did not experienced smallpox. However, she had had cowpox before. It's a different infection, but there are similarities in the viruses that may cause them. So Jenner collected some samples from the milkmaid and inoculated it into the boy James. Okay? So true enough, okay, although James were playing with his sick friends or carrier friends, but uh, he did not pray victim to smallpox during his time. Now moving on to 19th century. 19th century is considered the era of public health. Here, people were more appreciative of cleanliness. People were more understanding of the significance of proper hygiene. Water treatment has been established and put up, and people are now pasteurizing beverages such as milk and wine before they sell it out. So there has been so many milestones during this era. One of the major contributions in the 19th century medical science is Rudolf Virchow. He is considered as the father of microscopic pathology and contributed to procedures in autopsy, yeah, and he founded the archives of pathology in Berlin. Then we have Hermann von Felling. Felling performed the first quantitative test for urine. The test he created is a test for reducing sugars, the Felling's test. We actually still perform that in our biochemistry laboratory. Then we have John Snow. Okay, so John Snow studied cholera outbreak in London during his time and brought the situation under control quite successfully. You might ask how. He was a really curious individual. He had that detective blood in him. So he tried to uh, create a map to trace, and identify the locations of those who were sick with cholera. And so this was the map he created. And then he went and visited them. He went house to house and interviewed the patients and asked them about their food handling. How did they prepare their food? How did they cook their food? Where did they get their water? Where did their drinking water came from? And so he discovered in his interviews that most, if not all, of those that suffered from cholera at that time collected water from a pump in Broad Street. And this is the actual pump. It was preserved. Okay? And so, because he was able to discover that this pump is actually the source of the contamination, they sealed this off from public use, saving the country from so many deaths. Then another great contributor in the 19th century is Louis Pasteur, a French scientist who contributed to the world the procedure or the technique of pasteurization. He developed anthrax vaccine, and he also instigated the germ theory of disease, or created, rather, conceptualized the germ theory of disease. Pasteurization came about because a lot of the wine merchants had problems about their wine turning sour. So it's not wine anymore, it's vinegar. So customers complain that it's sour. So when Louis Pasteur looked into the situation, he found out that the wine turned sour because of the bacterial fermentation of the sugar in the wine, producing acetic acid, turning the wine into vinegar. So he experimented on different procedures that will kill the microbes, most, if not all of the microbes, but still maintain the taste, the texture, and the smoothness of wine. And he was able to do that successfully, heating wine at very low temperatures to kill microorganisms. Okay? And after that, that procedure known as pasteurization is practically used in all of the beverages that are sold in the market. Okay, now the germ theory of disease that he conceptualized stated that a single microorganism causes a particular disease. Uh, Unlike the common concept at that time that there's only one great causative agent that causes all types of infections. So he does not believe that. However, he did not have the proof and the evidence just yet to for his germ theory of disease until much later when Robert Koch came about. Okay, so Robert Koch actually believed the germ theory of disease conceptualized by Louis Pasteur. And so he experimented also on his own. He tried to uh, experiment on a dead rat, a dead mouse, rather, and collected blood sample from that uh, animal. The blood sample was examined microscopically, and whatever he saw, he noted that in his log books. And then the rest of the samples were inoculated or planted on gelatin media to observe growth parameters. And then he injected, okay, the organisms or whatever was growing on the gelatin media into a healthy mouse or animal of the same species. Okay, so when the mouse died, he again collected blood from that animal and did the same steps he did previously to the first animal. So he collected blood sample, examined the blood sample microscopically, and then recorded whatever he saw, and then the rest of the samples were planted. Now, he also observed the growth characteristics that he saw on the first gelatin media for the first animal and compared it to the growth characteristics of the organisms on the second gelatin media from the second uh, dead animal. And because of that experimentation, he was actually able to see that whatever caused the death of the first animal also caused the death of the second animal, primarily because of the similarities in structures and the similarities in the growth characteristics. However, okay, Cox postulates is limited to a certain type of situations. It cannot be used, for all types of situations. Okay? Like for today, there are a lot of infections that are caused by different types of microbials. So we call it polymicrobial infections or super infections. Now moving on, let's look into the history of medical technology in America or the United States. Okay, so looking at the illustration, you'd see, okay, uh, that the scientists here used microscopes with a single eyepiece. Okay, here the miss, the scientist, is actually inoculating loop using a very large Bunsen burner. And the other scientist here on this side tried to siphon chemical from the bottle using rubber tubing. That's how they did that at that time. Okay, so the late 19th century America saw the emergence of clinical laboratories. So a lot of clinical laboratories were put up during the late 19th century. The first one was created by Dr. William Welch. He established a laboratory at the Bellevue Hospital Medical College. This was followed by a clinical laboratory put up by Dr. William Osler at the Johns Hopkins Hospital. Okay, so there they performed routine exams, primarily urinalysis and blood examinations and stool examinations. And they also had special procedures such as identification of blood malarial parasites. Okay. Then it was followed suit by the establishment of the William Pepper Laboratory in the University of Pennsylvania. Later, Pennsylvania released a state legislature that required all hospitals to have adequate laboratory and employ a full-time laboratory technician. 1923, the University of Minnesota first offered a degree level program for medical technology. And then by 1936, the American Board of Pathology was established. Okay. World War I showed an upsurge in the number of clinical laboratories in the area, but World War II had a great effect on laboratory medicine. The result of the war uh, caused the development of closed system blood collection, meaning they were now using blood bags instead of open blood collection or from person to person. Then instrumentation became advanced. There was automation in the laboratory, so we had machines now at that time. And then, of course, quality control programs to check from time to time whether or not accuracy and precision is observed in the laboratory. Now, moving on to history of the medical technology in the Philippines. Okay, so medical technology, or clinical laboratories for that matter, came about in the Philippines because of the 6th Infantry Division of the US Army. When they came here, they brought with them their medical staff and established the 26th Medical Laboratory of the 6th US Army. So that was the actual first clinical laboratory in the Philippines. Okay, now it is known as the Public Health Laboratory, which is a part of the Manila Health Department. June 1945, the staff of the U.S. Army, the laboratory staff of the U.S. Army left, and they endorsed the clinical laboratory to the National Department of Health. Later on, a Filipino medical staff of that laboratory, Dr. Pio de Roda, preserved the lab with the help of his friend, Dr. Mariano Icasiano. So now that laboratory is known as the Manila Public Health Lab. Later, Dr. Pio de Roda asked the help of Dr. Prudencia Santa Ana to create a program for lab workers. So they conducted a training program for aspiring lab workers, but this ended in 1954 when the Bureau of Private Education approved a 4-year course in BSMT. And what is this? Where is this? Okay. That 4-year course that was approved was established by Dr. Willa Hilgert Hedrick. She is considered as the founder of medical technology education in the Philippines. She is actually a Seventh-day Adventist missionary in the Philippines, and she created the program in partnership with the laboratory at the Manila Sanitarium and Hospital. Okay, so she opened the first school of medtech in the Philippines. 1954, the Manila Sanitarium Hospital School of Medical Technology was transferred to a sister institution known as the Philippine Union College, or PUC, in Baisa, Caloocan City. Now it is known as the Adventist University of the Philippines. The first graduate of that program was Dr. Jesse O'Malley. Okay. In 1957, UST offered an elective course in pharmacy that would lead to the degree of medical technology. And then by 1961, BSMT program was already recognized. So that is the glimpse, our glimpse into the past of medical technology. So I would leave you with saying by Pearl Buck that if you want to understand today, you have to search yesterday. Thank you for listening.